Today, we are going to demonstrate that plants, in fact, do need carbon dioxide in order to go through photosynthesis. So in this experiment, you are going to use spinach leaves, which Liz is demonstrating for us, and then you are going to poke holes in it, basically, or use a hole puncher and pull out these little discs. Those discs, you're going to do 10 of them for water and 10 of them for our sodium bicarbonate solution. Then you're going to put them into these cute little syringes, and you're going to run them for about 10 to sometimes 20 minutes to see what happens to the spinach leaves. So after you poke the holes, okay, really, you're making a disc with a hole puncher.
And we're avoiding the veins.
Yes.
There's no photosynthetic material, or limited, right? No photosynthetic.
There's less of the chloroplasts in that area. It's true.
So we would have 10 of these.
Yeah, so you'd have 10.
2 of these.
Yeah, we're keeping it simple today, folks.
Yeah. So we've got 2.
So we put that in and then fill it up with whichever solution that you are measuring. So this one says sodium bicarbonate on it. Now, sodium bicarbonate is basically baking soda. It's baking soda, and we've dissolved it into water. This is going to dissociate in water to allow carbon dioxide to be available for the spinach leaf to actually go through photosynthesis. All right, so we are going to suck some of that sodium bicarbonate into the syringe. All right, and then you are going to have to actually kind of use a vacuum system to get the discs to float down. So if you can look closely, whoa. Inside the syringe, you can see that the discs are floating towards the top because right now they are filled with air. So in order to vacuum that out, basically, you are going to follow a very sophisticated method where you are going to plug the end with your thumb and then pull on the syringe. As you do this, you're going to shake, and twist it around a little bit, all right, and then you are going to release the plunger and release your thumb, all right, and then you will see, we're a little bit out of screen here, that at the top there's a little bit of air. That air needs to be pushed out. So it's just water and bicarb, so like make a mess. I always say this is the part where you're a bad nurse in a movie that flicks exactly. All right. And then you're going to do it again, right? So you're going to do this again and again and again until all the discs sink to the bottom. So you can see even on just that one pull, the spinach disc is moving down. Sometimes this is a little more challenging, all right? But you might have to do it a couple of times. So you'll do this until all 10 discs have sunk to the black part on the plunger. Then you're going to take both your sodium bicarbonate syringe and your water syringe, and you're going to set it up in this very fancy apparatus. You are going to see that we have a light. Ooh, should we turn it on?
Good idea.
All right, so what are the things plants need for photosynthesis? They need carbon dioxide, right? So we have given that to them with our sodium bicarbonate. They also need water, which is, well, in the solution for the sodium bicarbonate. And they need light. So by setting up this apparatus, we're giving the plant light, the carbon dioxide with the sodium bicarbonate, and then water. Now you'll also notice that we are running a syringe that is just water. So what is that missing?
The sodium bicarbonate. That's carbon dioxide.
Which means that those spinach leaves are not going to have as much carbon dioxide available to them as the sodium bicarbonate. The other thing you're going to notice about this slightly silly apparatus is that there's a giant fishbowl in the center. So fishbowl. Liz, why is the fishbowl here?
The fishbowl is here to act as a heat sink because although the light is coming from the light bulb, there's also heat that's coming from the light bulb, which could affect our experiment.
Because if it gets too hot, what's going to happen to those proteins?
They are going to denature.
And if we denature, we can't do photosynthesis or anything, dead.
Right. Enzymes are important in order to allow for plants to do the process of photosynthesis.
So if we look at, so Liz and I set this up earlier so that you can see what would happen at the end. Can we turn off the
We
light?
can turn
I think
it
that's
off.
going to be best. Oh, wow. Okay, there we go. So what you will notice is that the syringe with the sodium bicarbonate provided carbon dioxide, water, and then the light from the light. And all of the spinach leaves rose to the top because they were producing a very important gas. What is made during photosynthesis?
Oxygen.
Yes. So the spinach leaf was making its sugar during photosynthesis and then oxygen. The oxygen gets kind of stuck in the leaf disc and then it floats to the top. So during this experiment, we're really testing how much oxygen is produced to record how much photosynthesis happened. You will also notice in this water one, every single one of the water discs is still sitting down at the bottom. They did not rise at all because what didn't we give them?
Carbon dioxide, the sodium
Yes.
bicarbonate.
So, simply showing that plants need carbon dioxide in order to go through photosynthesis. Without it, nothing can happen. And the spinach just sits there sad at the bottom.
So if we think about this experiment, Marcia, right? If we think about independent.
Oh
variables,
man, that was a long
right?
time ago.
That was a long time ago. That would have been week one.
So what did we set? What was our independent variable? What did we get to control? Sorry, the great production value of the CLC biology team.
We're getting an official gig after this.
Oh
one.
yeah, I feel like I deserve lots of money.
Steven Spielberg is going to come knocking.
So we were able to control how much carbon dioxide the plant got, right? So our sodium bicarbonate versus our no sodium bicarbonate is our independent variable. And then what did we measure at the end, or what was our dependent variable? That would be terrible.
Oxygen.
The oxygen that was produced, or as we measured it, how many discs were able to rise to the top because of the amount of oxygen that they produced.
Things that we standardized, right? Light.
Light and heat,
And
right? We also standardized how many discs we put into each syringe, right? There were 10 in each.
We use the same organism, right?
We also use approximately the same amount of water and sodium bicarbonate, right? So our syringes are equal. That was a standardized variable. And I think that is the story of how plants use carbon dioxide in order to make photosynthesis happen.
Yep. Good work.